Fertilization in humans. Fertilization is a process of combining the male gamete sperm with the female gamete egg. The product of fertilization is called a zygote. Gametes involved in fertilization. The gametes involved in fertilization are egg and sperm. Egg. Eggs are large, symmetrical and non-motile cells. Human eggs are arrested in the metaphase of the second meiotic division and complete meiosis only upon fertilization. Their surface is covered by microvilli. Eggs are surrounded by zona pellucida, which is a glycoprotein coat composed of 3 glycoproteins. All 3 of glycoproteins contain O and N linked oligosaccharides. The zona pellucida is not an osmotic barrier, however, it is the barrier to the sperm. Sperm. Sperm are small, asymmetrical and motile cells. They have 3 compartments: head, midpiece and tail. Head. The head portion is mainly a cell nucleus. It consists of genetic substances called chromosomes. The head of the sperm is covered by a cap known as the acrosome. Midpiece. It is at the proximal portion of the tail. It contains a sheath of mitochondria. It produces ATP necessary for the beating of the tail. Tail. It is also called as the flagellum. It is a slender, hair-like bundle of filaments that connects to the head and the middle portion. Steps involved in fertilization. Approach of the spermatozoon to the egg, the reaction of the egg, the essence of activation, the spermatozoon in the egg interior, changes in the organization of egg cytoplasm caused by fertilization. First, approach of the spermatozoon to the egg. After both the eggs and the spermatozoon are discharged into the surrounding medium, or after the spermatozoa have been introduced into the genital ducts of the female, in the case of internal fertilization, the first step is the encounter of the spermatozoon and the egg. This encounter is brought about by the swimming movements of the spermatozoa. The egg is not accessible from every angle and the spermatozoa are directed to a certain opening which leads to the egg. A very fine chemical mechanism is involved in this process. The spermatozoa become sticky and adhere to the surface of the egg and even to each other. The mutual adhesion of the spermatozoa results in their clumping or agglutination. The substance causing agglutination of spermatozoa is called fertilizin. The fertilizin is continuously emitted from mature eggs and that is directly involved in the reaction between the egg and the spermatozoon. The mechanism of penetration is chemical. The spermatozoon reaches the egg and produces substances of an enzymatic nature known under the general name sperm lysins, which dissolves the egg membrane locally and make the path clear for the spermatozoon to reach the surface of the egg. The sperm lysins are produced presumably by the acrosome of the spermatozoon. The production of sperm lysins is thus one of the aspects of the secretory activity of these bodies. The situation is complicated by the presence around the egg membrane, the zona pellucida, of a layer of follicle cells, the corona radiata, which the egg carries with it when it leaves the ovary. The spermatozoon has to pass through this layer on its way to the egg. The sperm produce an enzyme, hyaluronidase, which dissolves the mucopolysaccharide, hyaluronic acid, the substance cementing the follicle cells together. The enzyme thus enables the spermatozoon to make way for itself through the corona radiata. Under the influence of the substances diffusing from the eggs, the structure of the sperm changes. The main change concerns the acrosome, the peripheral part of which collapses and becomes extruded and partially dissolved in water. The central part of the acrosome elongates and transforms into a long, thin filament and possibly a tube which has considerable rigidity and protrudes forward from the head of the spermatozoon. This structure has been called the acrosomal filament. The sperm, which have developed acrosomal filaments, are considered to have been activated and are ready to penetrate into the egg. As the sperm approaches the egg, the acrosomal filament is pushed through the jelly and through the vitelline membrane, the pathway for it being cleared by the action of lysins. Eventually, the tip of the acrosomal filament touches the surface membrane of the egg cytoplasm. The reaction of the egg. Immediately after the acrosomal filament of the spermatozoon touches the surface of the egg, the cytoplasm of the egg bulges forward at the point of contact, producing a process of hyaline cytoplasm, the fertilization cone. The fertilization cone gradually engulfs the spermatozoon, then begins to retract, carrying the sperm inward, surrounded by the hyaline cytoplasm of the fertilization cone. Even before the spermatozoon thus penetrates into the interior of the egg shows signs that it has become profoundly changed and that it has been activated. There are changes happening in the surface of the egg cytoplasm which are known under the general name of the cortical reaction. Immediately after fertilization, the two layers separate from one another and the outer layer separates from the egg as the fertilization membrane. At the same time, the cortical granules start swelling rapidly and explode. Bits of cytoplasm lying between the outer surface of the cortical granules and the plasma lemma may become disconnected from the egg cytoplasm and adhere to the fertilization membrane. The fertilization membrane, formed after the first minutes of fertilization, is soon reinforced by the denser lamellar substance released from the exploding cortical granules. The fertilization membrane thus becomes much thicker and stronger. In addition to strengthening the fertilization membrane and producing or helping to produce the fluid filling the perivitelline space, the cortical granules also give rise to a viscous hyaline layer which adheres closely to the surface of the egg, and during cleavage it helps to keep the blastomeres together. The jelly layer which surrounds the unfertilized egg persists for some minutes after fertilization but eventually dissolves without the trace. No new membrane is formed around the egg at the time of fertilization. The perivitelline space may increase slightly, not owing so much to the lifting of the membrane as to a shrinkage of the egg itself, which loses a certain amount of water to the perivitelline fluid. The cortical reaction of the egg, and in particular the lifting of the fertilization membrane, are defense reactions which prevent the penetration of additional spermatozoa after the first one has established contact with the egg. The essence of activation. The combining of the fertilizin of the egg with the anti-fertilizin of the sperm would then be the beginning of the changes in the physiochemical system of the egg, the key reaction of fertilization. The increase of oxidation rate cannot be considered as the key reaction of fertilization. Other metabolic changes found after fertilization in some, though by no means all animals, are production of considerable amount of acid during first several minutes after fertilization, increase of permeability of the egg membrane, increase in the activity of proteolytic enzymes. Before fertilization, the ribosomes in the egg cytoplasm are single. After fertilization, increasing quantities are joined in groups, which shows that they are actively engaged in protein synthesis. The spermatozoon in the egg interior. The whole spermatozoon head, middle piece and tail penetrates into the cytoplasm and for a short time may be seen lying intact in the interior of the egg. The middle piece appears to enter the egg cytoplasm in most cases. The mitochondria contained in the middle piece have been observed in some cases to scatter in the cytoplasm of the egg. The egg completes its first reduction division in the ovary and reaches the metaphase stage of the second meiotic division. In this stage, all further progress is arrested, ovulation takes place, and the egg may become fertilized. The second reduction division is completed and the second polar body is extruded only if the egg is fertilized by the spermatozoon or activated in some other way. When the spermatozoon first penetrates into the egg cytoplasm, it moves with the acrosome at its front. The other parts of the spermatozoon, if still discernible by this stage, lose connection with the nucleus or the centrosome. Both the nucleus and the centrosome change their appearance. The nucleus, which is now referred to as the male pronucleus, starts swelling, and the chromatin, which is very closely packed spermatozoon, becomes again finely granular. The sperm nucleus, together with the centrosome, moves through the egg cytoplasm towards the area where the fusion with the nucleus of the egg, the female pronucleus, is to take place. The trajectory of the sperm head may be marked by pigment granules trailing along its path. This is referred to as the penetration path. The two pronuclei actually fuse together, the nuclear membranes become broken at the point of contact, and the contents of the nuclei unite into one mass surrounded by a common nuclear membrane. The nuclear membranes in both pronuclei dissolve and the chromosomes release. The centrosome of the spermatozoon has divided into two and an achromatic spindle has been formed to which the chromosomes derived from the male and the female pronucleus become attached. Only after the completion of the first division of the fertilized egg do the paternal and the maternal chromosomes become enclosed by common nuclear membrane in the nuclei of the two daughter cells into which the egg has divided. Changes in the organization of egg cytoplasm caused by fertilization. In addition to activating the egg, the distribution of various cytoplasmic substances and inclusions in the egg at the beginning of cleavage may be very considerably different from that in the unfertilized egg, and even qualitatively new areas may sometime appear. As soon as spermatozoon enters the egg, the yellow cortical cytoplasm falls into violent commotion. First of all, the yellow cytoplasm begins to stream down along the surface of the egg towards the vegetal pole, and for a time it accumulates as a cap on the vegetal pole. 4 different kinds of cytoplasm are now present in the egg. During development, the different kinds of cytoplasm give rise to specific parts of the embryo. Only a few minutes after the sperm has penetrated the egg, the surface layer of protoplasm starts contracting towards the animal pole of the egg, and in the course of this contraction, the pigmented protoplasm covers up the gap in the pigment layer at the animal pole where the first polar body has been given off. The marginal cytoplasm, although bearing some pigment, is much lighter colored than the cortical cytoplasm. The result is that there appears on the dorsal side of the egg a subequatorial lightly pigmented crescentic area known as the gray crescent. The amount of yolk is the decisive factor in determining the position of the gray crescent. Any part of the marginal zone which lies opposite the point of entry of the spermatozoon may be transformed into the gray crescent. In respect to the development of its bilateral symmetry, the egg appears to have a very considerable amount of flexibility. Fertilization age. Fertilization age is also known as the gestational age. It takes the beginning of the last menstrual point, LMP. It is calculated by adding 14 days to fertilization age and vice versa. Fertilization usually occurs within a day of ovulation. The average time to birth has been estimated to be 268 days, that is 38 weeks and 2 days from ovulation. The fertilization age is sometimes used after birth as well to estimate various risk factors. Diseases affecting human fertility. The following are a few of the diseases that can occur and be present during the process. Polyspermy results from the multiple sperm fertilizing an egg, leading to an offset number of chromosomes within the embryo. Polycystic ovarian syndrome, condition in which the woman does not produce stimulating hormones and excessively produces androgens. Autoimmune disorders lead to complications in implanting of the egg in the uterus. Cancer ultimately affects fertility and may lead to birth defects or miscarriages. Endocrine system disorders affect human fertility by the body's ability to produce the level of hormones needed to successfully carry a zygote. Endometriosis, condition that affects women in which the tissue normally produced in the uterus proceeds to grow outside the uterus. Significance of fertilization. Fertilization restores the diploid number of chromosomes, that is 46 in human beings. It provides a stimulus for the ovum to complete its maturation. Fertilization combines the characteristics of two parents. This brings about recombination of genes and introduces variations. It determines the sex of the embryo in humans, and fertilization introduces the centrioles which are absent in the ovum.